All right, 8.2 cell respiration. How do we extract the energy from sugar to produce ATP to power basically everything in our bodies? All right, so things you should know about cell respiration. We're only going to talk about how to catabolize glucose, and to catabolize, like catastrophe, is to like blow it up and break it apart, as opposed to anabolism, which would be the opposite of catabolism. So, three important parts of this definition. It is the controlled release, which means we're going to go step by step, right? You could do this entire reaction by just lighting a sugar cube on fire all at once, but we are going to do step by step, systematically dismantling. Glucose is the only organic compound we're going to talk about, but you can burn other stuff like fat and protein. Um, in emergency situations, usually don't want to burn protein. And we're trying to make ATP, adenosine triphosphate, 3 phosphates. So, yeah, controlled release, step by step. Every single step is going to be catalyzed by an enzyme to help it happen faster and a lower temperature. And we can also shut it off by modifying if those enzymes are produced or inhibited. So, these enzymes are really nice for speed and selectability. A very important verb here is to oxidize. We are adding oxygen to sugar. Here's the overall reaction. You can see oxygen is a reactant. So this is an oxidation. There are two other ways to oxidize, but the easiest way to think about it is adding oxygen. So you can see how the carbon here is what's being oxidized. We have one carbon for every one oxygen in glucose. It's a 6 to 6, which is a 1 to 1 ratio. And at the end, we're getting carbon dioxide. So now we're going to have two oxygens for every carbon. So we're adding oxygen and oxidizing carbon. We're going to get water as a waste product. And, you have water everywhere in your body, so you don't even notice this water. But this CO2 needs to be breathed out because that's going to go in your bloodstream and make it acidic. So you need to breathe out this CO2 to get rid of it. Okay, again, we're only talking about glucose, and you should already be aware it is a hexagon with 6 carbons. You don't need to know all the positions of the oxygens and hydrogens. We're going to ignore oxygens and hydrogens for most of this process and just focus on the carbons because that is the element for life. Okay, we're only talking about glucose. It is a monosaccharide, right? If you eat starch or you have some glycogen, that could be 1,000 glucoses in a row. Your digestive system would cut that into little glucoses, and those glucoses would be put into your cells to be metabolized or catabolized. Yep. Glucose is so good at being burned because it's the only thing you can burn without oxygen, right? So if you don't have enough oxygen, let's say you're working out hard or you're underwater, glucose is the only way you can make some ATP. Okay, that's its formula. ATP is also super important. This entire hours of lecture that and studying you're doing is all to take this little phosphate right here and put it right there next to it. We're trying to form this third phosphate bond. And that's the entirety of the purpose of cell respiration. This is why you breathe, to put this little phosphate right there. That's it. Okay. Triphosphate, the important letter here is T for tri. That's 3 phosphates. So you can see here all 3 phosphates linked together. This would be like an all-purpose battery. An equivalent for you would be a battery that powers your cell phone, your car, and your house. This little molecule powers almost everything in your body. A verb you need to know is phosphorylation. That is adding a phosphate. So when we say phosphorylation during this process, what we're doing is we're phosphorylating ADP. Phosphorylate, add the third phosphate to make ATP. Okay, and then we're going to do the opposite. This would be called dephosphorylation, which you don't need to know, but that's the opposite. Is removing a phosphate is dephosphorylation. So this is why we make the phosphate, so we can take the third phosphate off. That's going to release some energy to power whatever process, right? Whether it be a muscle moving or the sodium potassium pump, so that you can think. Every process that you can think of requiring energy is probably powered by ATP. Okay, so once we've used the energy from ATP, we now have to re-energize the ADP. It's like a battery, right? You got to recharge the ADP low energy battery, turn on the high energy ATP useful battery. Okay, before we get into the process, some vocab terms you need to know. You need to be able to come up with this table by memory. So I call it the HOE table, HOE. And you need to know which one is which. So oxidation is the exact opposite of reduction. In fact, you can't have an oxidation without a reduction, because how are you supposed to lose hydrogen from one molecule if it has nowhere to go? So if one molecule is losing hydrogen, that has to be gained somewhere else. So these always happen in pairs. The way that I make this table from memory myself is oxidation is a gain of oxygen. So I start with this cell here, that oxidation is a gain of oxygen. And then from there, I just have everything be the opposite. Oxidation is the opposite of reduction. So these are opposites. Oxygen is going to be the opposite of hydrogen. So these are opposites. And then for electrons, I use, you can use a couple techniques. The one that I like is Leo the lion says grr. So Leo, loss of electrons, is an oxidation. Grr, gain of electrons, is a reduction. You might also use oil rig. Oxidation is a loss of electrons. Reduction is a gain of electrons. Yeah, either way. Okay, so again, if something is reduced, we're going to add an H to this molecule called NAD, turn into NADH. If NAD is being reduced, glucose is being oxidized. Okay. Yeah, so the mnemonics I just talked about here. Grr. Right, so here this is a gain of hydrogen, so that would be reduction. This is also a gain of hydrogen, so that would also be reduction. Okay, there's also going to be a gain of electrons here to hold the hydrogen to it, so it's a reduction for two reasons. You're also gaining electrons. What about the carbons here? We're going from, this is what I mentioned earlier, we're going from 1 to 1 to 1 to 2. Now there's more oxygens per carbon, so that's an oxidation. This is the opposite of this first process. So if this was a reduction, this must be an oxidation, because we're losing hydrogen. Okay, a lot of acronyms here. Abbreviations. You already know what ATP is. And then we have these other two energy carriers. So you need to lump these three in your mind, because these are all high energy carriers. They're going to be made and be very useful to power something. Okay. ATP powers like everything, and it powers something by losing a phosphate. NADH and FADH are much more specific. They power one thing and one thing only. They power the ETC, which is going to be step 4 of cell respiration. So the whole purpose of taking in this sugar and burning it, we're not going to directly make ATP. We have to make this little intermediate called NADH. So we're going to burn the sugar, and you'll see in the first 3 steps of cell respiration, we make more NADH than anything else. We might make some ATP, but the NADH is really what we're after from the first half of cell respiration. And then in the last half of cell respiration, we're going to use the NADH we just made, use it immediately, and power the ETC, and that's going to make 90% of all your ATP. So you might be like, why don't we just oxidize the sugar and directly make ATP? Well, that's not how it evolved. It evolved to have this little intermediate made and broken or used. Okay, so that's just how it evolved. All right, if you're wondering what these molecules look like, this is ATP. We've already talked about that. Those are the 3 phosphates here. If you're wondering, FADH looks like this, dinucleotide. It's got like 2 nucleotide looking things on here. And then NADH also has 2 kind of nucleotide looking things here with a sugar, phosphate, nitrogen base, but you don't need to know these pictures. Okay, does not cause a net release of energy. So this would be A, right? This would have to take in energy. You're getting a low energy ADP, turn it into a high energy ATP. Okay. All right, so as we get into the 5 steps, the GLKEC of cell respiration, we need to keep track of how many carbons. And we're going to use this shorthand 6C for 6 carbons, 3C. We're going to keep tearing carbons apart, and CO2 is the final resting place of those carbons. Okay, so we're going to lose carbons one at a time as CO2, and then that goes to your lungs to be breathed out. Keep track of those 3 high energy compounds. We're going to make some ATP in the beginning. We're going to make mostly NADH in the beginning and a little bit of FADH too in there, which does the exact same thing as NADH. These are cousins. They do the exact same thing. Okay, this is kind of weird. We're not going to use oxygen until the very last step. You know how important oxygen is, and all of the oxygen you breathe is just for cell respiration, nothing else. So you might be like, oh, it must be used in the beginning if it's so important, but it's actually not going to be used until the very last step. Okay. We also need to keep track of the location. Not all of cell respiration happens in the mitochondria. It's not just the powerhouse of the cell. We're also going to tear apart glucose in the cytoplasm. Okay, we have 2 types of cell respiration. Anaerobic, an means not. So this is the one that's not no oxygen. That's going to be a little bit of ATP. Without oxygen, you can't use the mitochondria. So it's all in the cytoplasm. This is an emergency situation. We just need to get as much ATP as we can. We're not going to get enough. Okay, the 2 steps of anaerobic are glycolysis, then fermentation. Okay, GF. Aerobic respiration, this is the big one. This is what we'll spend 75% of this lecture on. 5 steps, GLKEC. So come up with a mnemonic for that. The one that I like is good luck kissing or kicking every cat. That's kind of a fun one. So glycolysis, the link reaction, Krebs cycle, electron transport chain, which I'll usually call ETC, and chemiosmosis, which is kind of a stupid word. All right, here's the picture. Here is the GF of anaerobic. So if you don't have oxygen, notice none of these arrows are happening inside the mitochondria. These are the 2 steps of anaerobic. If we do have aerobic, then that's everything. That's all 5 of these steps. GL, so this is glycolysis, GLKEC. Glycolysis, link reaction is this little arrow here. It's not labeled. Krebs cycle, electron transport chain, and then chemiosmosis is this arrow that's not labeled, where you make 90% of your ATP. Notice this ATP is bigger than these other 2. Okay. Ooh, what did I do? Okay. So, the odds of glycolysis being on your test are very high because both aerobic and anaerobic always start with glycolysis. GF for anaerobic, glycolysis fermentation, and then GLKEC for aerobic. Both start with a G. Glyco means glucose. Lysis means split. We are splitting glucose. This happens in the cytoplasm. You don't even need, if you could survive from glycolysis, which some bacteria can, you wouldn't need to breathe, because this doesn't need oxygen. So the whole process, we're going to make 2 of everything. We're going to take a 6-carbon compound and we're going to exactly split it in half. Notice no CO2 is made. We haven't lost any carbons as gaseous CO2. We're just taking a 6th carbon, splitting it in half into 2 carbons. We're going to energize 2 low energy NADs into NADH, and we're going to energize 2 ADPs into 2 high energy ATPs. So just remember 2, 2, 2. And we're making our carbon compound and 2 of the 3 energy carriers. Notice in all 3 of these first 3 steps, we're going to make more NADH than anything else, or it's going to be tied for the most. So we got as much NADH as ATP. Make 2 of everything. Okay, where do these reactants come from? Glucose from your food. NAD comes from using NADH. So we need NADH to get used up to regenerate our NAD. Otherwise, we'll run out of that. And then you're never going to run out of ADP. There's always something for ATP to power. So the NAD is actually the biggest issue that we might run out of. Okay, one more thing you need to know about glycolysis is we do make 2 ATPs by the end, but it's a 4-2 situation. So we actually use 2 ATPs in the first couple steps. It's actually a 10-step process. But we use 2 ATPs in the first couple of steps. And by the end, we're going to make 4. Okay, so that's a net production of 2. If you remember the term, if you've ever heard the term gross and expenses, like economics for a business, your gross is 4. Okay, you make a total of 4, but you've used 2 is like your investment as your expenses. So that's a net profit of 2 ATPs total. Okay, so you need to know 2 ATPs are used, 4 ATPs. This reaction here is just the overall. Overall, you make 2 ATPs because you actually make 4, but two get used in the beginning. Okay? So we're going to add some phosphates to the glucose with this energy investment phase of using the two ATPs. Then they're going to split, and then we're going to oxidize, and that's going to make it a lower energy pyruvate molecule. We're going to extract some of that energy from oxidation to make our NADH and ATP. Okay. So now there's the fork in the road. We've just done glycolysis. We could still do anaerobic because anaerobic still does glycolysis in the beginning, just like aerobic. If there is oxygen, we're going to take these 3 carbon pyruvates. The way I remember pyruvate has 3 carbons is because it has pi, and pi, the constant is 3.14 whatever. So I just remember pi 3 carbons in pyruvate. That's a number you need to have memorized. So if there is oxygen, we still have more carbon-carbon bonds that we can deal with and break and make a bunch of CO2 out of. If there's no oxygen, this pyruvate, we are done oxidizing the pyruvate. We can't make any more ATP. So that's what we'll talk about next. If there's no oxygen, all we have is glycolysis for your ATP. We make 2 ATPs per glucose for glycolysis, so that's something. Not enough to keep you alive, but that's something. And remember those 3 reactants, glucose, ADP, and NAD. So if all we can do is glycolysis because we're out of oxygen, then we better make sure we have all 3 of these starting materials, okay? We're assuming you've eaten recently, so you have some glycogen and glucose, and you're not going to run out of glucose. You're never going to run out of ADP. There's always something for ATP to power and turn back into ADP, but you will run out of NAD, okay? We need NADH to turn back into NAD, and normally that would happen in the mitochondria. But if we don't have oxygen, NADH cannot power the ETC. It can't use its energy usefully. So what we have to do is fermentation. Okay, so we are going to take pyruvate, and fermentation is really weird. In every step of cell respiration, we usually oxidize the sugar so that we can make NADH, so that we can reduce NAD. But in fermentation, we're actually going to reduce the sugar because we need NADH to do something. We need to turn the NADH back into NAD. That H and those electrons need somewhere to go. If we're going to oxidize NADH back into NAD, something needs to be reduced. And so that something is going to be pyruvate. And as we reduce it into its waste product, in humans, we make lactic acid. Notice it has still 3 carbons, so we haven't lost any carbons. In yeast, they actually do make some CO2 by fermentation. So the pyruvate is going to lose one of those carbons as CO2. And if you do the math, we had a 3 carbon. We've lost 1 carbon. Ethanol actually has 2 carbons, which you don't need to know unless you're in chem. But yeah, so that's we use this ethanol to make alcoholic beverages. We use this CO2 in bread dough to make it rise. Okay, so that's anaerobic. All right.